Today's discussion delves into a fascinating area of research, the intersection of lifestyle choices and brain health. We'll be exploring the neurotrophic effects of intermittent fasting, calorie restriction, and exercise. It's a complex topic, but one with potentially life-changing implications.
Precisely. The brain's ability to adapt and regenerate throughout life is a remarkable process, and understanding how these interventions influence it is crucial for promoting healthy aging and mitigating neurodegenerative diseases.
And the research suggests these seemingly simple lifestyle changes can have profound impacts on neurogenesis, synaptic plasticity, and neuroprotection. It's not just about feeling better, it's about preserving cognitive function.
Absolutely. We're talking about the creation of new neurons, strengthening existing connections, and protecting the brain from damage. These are fundamental processes for learning, memory, and overall brain health.
So let's dive in. What are some of the key mechanisms through which these lifestyle interventions work?
A central concept is the metabolic switch from glucose to ketone bodies as the primary fuel source. When glucose is scarce, as during fasting or intense exercise, the body shifts to using ketone bodies, primarily beta-hydroxybutyrate, BHB.
And this metabolic shift isn't just about providing alternative fuel, it triggers a cascade of cellular events.
Exactly. This metabolic stress activates and inhibits various signaling pathways crucial for neurotrophic effects. Think of AMPK, mTOR, and sirtuins. These are key players in cellular energy regulation, stress response, and longevity.
So the body's response to this metabolic challenge is what drives the beneficial changes in the brain.
Precisely. It's the repeated cycling between glucose and ketone body utilization that seems particularly beneficial, promoting cellular regeneration and protection.
Let's talk about those signaling pathways in more detail. You mentioned AMPK, mTOR, and sirtuins. Can you elaborate on their roles?
Certainly. AMPK acts as a cellular energy sensor, activating pathways that conserve energy and promote metabolic efficiency. mTOR, on the other hand, regulates cell growth and protein synthesis. Sirtuins are a family of enzymes involved in DNA repair, stress resistance, and inflammation.
And how do these pathways interact with each other?
It's a complex interplay. AMPK often inhibits mTOR, while sirtuins are activated by increased NAD+, a molecule whose levels are influenced by both AMPK and the metabolic switch. These interactions are crucial for the neuroprotective effects.
So it's a finely tuned system, and these interventions help maintain that balance.
Precisely. They create a state of mild metabolic stress that optimizes these pathways, promoting neurogenesis, synaptic plasticity, and neuroprotection.
Let's break down the specific neurotrophic effects. How do these interventions impact neurogenesis, neuroprotection, and synaptic plasticity?
Neurogenesis, the formation of new neurons, is enhanced by these interventions, particularly in the hippocampus, a brain region crucial for learning and memory. Neuroprotection involves safeguarding neurons from damage, and synaptic plasticity refers to the brain's ability to adapt and reorganize its connections.
And how do these three processes work together?
They're interconnected. New neurons integrate into existing circuits, enhancing synaptic plasticity and contributing to improved cognitive function. Neuroprotection ensures the survival of these new and existing neurons, maintaining the structural and functional integrity of the brain.
So it's a synergistic effect where each process reinforces the others?
Exactly. It's a holistic process where the combined effects of neurogenesis, neuroprotection, and synaptic plasticity contribute to improved brain health and resilience.
Let's focus on exercise. How does physical activity contribute to these neurotrophic effects?
Exercise induces a metabolic challenge, triggering the glucose-to-ketone switch and activating AMPK. It also increases the production of BDNF, a key neurotrophic factor.
But exercise also produces reactive oxygen species, ROS, which can be damaging. How does the body manage this?
It's a matter of balance. Moderate exercise increases ROS, but the body's antioxidant defenses are also upregulated, leading to a beneficial adaptation. This is often referred to as hormesis, a mild stressor that strengthens the system.
So it's not just the intensity, but also the type and duration of exercise that matters.
Absolutely. The optimal type, intensity, and duration of exercise will vary depending on individual factors and the specific neurotrophic effects being targeted.
Now let's move on to intermittent fasting. How does this dietary approach influence brain health?
Intermittent fasting, like exercise, induces a metabolic challenge, leading to the glucose-to-ketone switch and activation of AMPK and sirtuins. This promotes cellular repair and reduces inflammation.
Are there different types of intermittent fasting and do they have different effects?
Yes, there are several approaches, including alternate day fasting, time-restricted feeding, and periodic fasting. The optimal approach will depend on individual preferences and tolerance.
And what about the potential downsides of intermittent fasting?
It's crucial to approach intermittent fasting cautiously, ensuring adequate nutrient intake and avoiding malnutrition. It's not suitable for everyone, and individual responses can vary.
Let's discuss calorie restriction. How does it compare to intermittent fasting?
Calorie restriction involves a consistent reduction in overall caloric intake, while intermittent fasting involves cycling between periods of eating and fasting. Both approaches induce metabolic stress, but they differ in their implementation.
And what about calorie restriction mimetics, CRMs?
CRMs are compounds that mimic some of the beneficial effects of calorie restriction without requiring a reduction in caloric intake. Resveratrol and rapamycin are examples of CRMs that have shown promise in preclinical studies.
But the effectiveness of CRMs has been questioned in some studies. What's the current consensus?
While some studies have shown limited efficacy in humans, the potential of CRMs in preventing and treating neurodegenerative diseases remains an area of active research. More robust clinical trials are needed to fully assess their effectiveness.
How do these interventions interact with epigenetic changes associated with aging?
Epigenetic modifications, changes in gene expression without alterations to the delay sequence, play a significant role in aging and age-related diseases. These interventions can positively modulate epigenetic marks, counteracting some of the detrimental effects of aging.
So these lifestyle changes can actually reverse or slow down the aging process at a molecular level.
While not a complete reversal, these interventions can significantly influence the aging process by modifying gene expression and cellular function, promoting a healthier and more resilient state.
How can these interventions be applied to the prevention and treatment of neurodegenerative diseases and depression?
The evidence suggests that these interventions can help mitigate the risk and progression of neurodegenerative diseases like Alzheimer's and Parkinson's disease, as well as major depressive disorder. They do this by promoting neurogenesis, neuroprotection, and synaptic plasticity, counteracting the cellular and molecular mechanisms underlying these conditions.
Are there specific recommendations for individuals considering these interventions?
It's crucial to consult with healthcare professionals before making significant changes to diet or exercise routines. Individual needs and health conditions must be considered to determine the most appropriate approach and to avoid potential risks.
What are some of the key research gaps that need to be addressed?
More research is needed to compare the effectiveness of different interventions, determine optimal dosages and durations, and investigate the interactions between different signaling pathways. Robust clinical trials with larger sample sizes are also crucial to confirm the findings from preclinical studies. Furthermore, the use of advanced techniques like metabolomics and live cell imaging can provide a more comprehensive understanding of the complex molecular mechanisms involved. So, in conclusion, the evidence strongly suggests that intermittent fasting, calorie restriction, and exercise can significantly impact brain health by influencing neurotrophic factors and signaling pathways. While more research is needed, these lifestyle interventions hold considerable promise for promoting healthy aging and mitigating age-related neurological disorders. Thank you for this discussion.